Welcome back. We are starting with the chapter 9. This will discuss about the antimicrobial chemotherapy. So first we are going to talk about that how the antimicrobial chemotherapy evolved from the antisepsis effort over the period of time. So the very first thing that the chemical agents were used to treat different types of disease and destroy pathogenic microbes or inhibit their growth within the host. And these were all observed by many scientists long time before even the first discovery of the antibiotics. And we're going to talk about those things. And the chapter will mainly talk about the antibiotics because these are the numbers which are against the bacteria. The number of antibiotics are far more than the combined number of the other, which are the antivirals, antifungal, antiprotozoal, and also even anti-helminth. Anti-helminth we are not going to consider in this chapter, but we are going to talk about the others along with the antibiotics, which is against the bacteria. In addition, we are going to talk about the antifungal, antiviral, and antiprotozoal as well. So microbial products are the derivatives that kill the susceptible microbes or inhibit their growth is known as the antibiotics. But most of the antibiotics are synonymous that they kill the other bacteria. And this is important. We do not use the term antibiotics if it is an antiviral, antiprotozoal, or antifungal. So let's take a look at some of the histories. German scientist Paul Ehrlich saw, and he was fascinated that some of the specific dyes bind to microbial cells, whereas they do not bind to the human or animal cells. And from that specific observation, he deduced and developed the concept of selective toxicity. And then what happens, he also identified the dyes that effectively treated African sleeping sickness, and this is caused by a protozoa, not a bacteria, which is a trypanosoma. But also, along with him, there was another scientist, a young Japanese scientist, Sakahiro Hato. He worked with Elric and identified the arsenic compound that can effectively treat syphilis. If you read your chapter opener, there is a good write-up about the syphilis, and many do think that in the Americas, the syphilis was not there. And when Columbus discovered America, from the European continent, the syphilis came to the New World. But some of the other people do argue that it was in there, but it was not treated or identified as a syphilis. They used to think that is a kind of a leprosy. But apart from that argument, the syphilis is caused by a spirochete. It is known as a Treponema pallidum. And Ehrlich and Hato discovered a compound which is derived from the arsenic known as arsfamine, and they named that, or commercially that was available under the name as salvarsan. So salvarsan is probably the first chemotherapeutic agent. And after that, what happened, Gerard Domac discovered the prontosil, which is a red leather dye. And he actually saw that that specific dye can protect the mice against streptococcus and staphylococcus infection. After that, Domac and then along with the Jacques Thorez-Truffaut, they all discovered these sulfur drugs or sulfonamide. And we are going to talk about that in much later in the chapter. So first, the penicillin, also we'd all know that that was the first antibiotic to be discovered. This was actually discovered by Ernst Duchesne in 1896, but this discovery was lost. It was not identified much later, and it was again by an accident where there was some staphylococcus plate that Alexander Fleming went to a vacation, and after coming back, he saw that there is some mold growth on that plate, and around that, there is no growth of any bacteria or the Staphylococcus aureus. So he reasoned that probably this particular mold, which he identified as Penicillium notatum, is releasing a compound which is being diffused, and that is killing that particular bacteria, which was Staphylococcus at that time. So he did not think he could develop further. But again, what happened, there was a good number of scientists at Oxford in 1939, Florey, and Heatley, they identified this compound, which Alexander Fleming named as penicillium. They isolated that compound, and then they were offered Nobel Prize in 1945. So that was the discovery of the penicillin. So after that, that streptomycin is an antibiotic active against the tuberculosis in that time, although it is not used today. That was done by Selman Waksman. And he was also awarded Nobel Prize in 1952 for this discovery. Then in 1953, chloramphenicol, neomycin, oxytetracycline, and the tetracycline were isolated. Most of these were isolated from the Streptomyces genus of the bacteria. So let's take a look at some of the general definition of this antimicrobial drug, that how we actually study. The most important thing that we have to understand, that when we develop a drug, the ability of the drug to kill or inhibit a pathogen while damaging the host as little possible, which is known as a selective toxicity. So this is one of the most challenging way, that you have to kill the pathogen but without harming the host. And we'll come back to that how we achieve this. So the next definition, therapeutic dose, is a drug level required for the clinical treatment of the disease. And then another toxic dose is the drug level at which the drug becomes too toxic for the patient or produces the side effect. Then we do have a specific index, we call this therapeutic index, or TI. This is the ratio of the toxic dose to the therapeutic dose. So this index is very important. What we mean by that, if there is a good difference in between the therapeutic dose and the toxic dose, then the therapeutic index is very high. What do we mean by that? Say, I'm just giving an example, that if a drug is good at 1 milligram per ml concentration, therapeutic dose is 1 milligram per concentration. On the other hand, the drug is toxic at the level of 10 milligram per concentration. So you have a gap in between 1 and 10, and it's a good therapeutic index. On the other hand, think about that. If a therapeutic dose of a drug is 8 milligram per ml, whereas the toxic dose remains the same as 10 milligram per ml, then the ratio is much less, because you just have a buffer of about 8 to 10, which is 2 milligram per ml, and that is a relatively much more dangerous drug. So this is how we classify the drugs which is selectively more toxic, having a high therapeutic index, are preferred compared to the low therapeutic index. But sometimes there are some drugs, even if they have side effects, we have to use in certain condition that we are going to discuss later in the chapter. So the side effects are the undesirable effects of the drug on the host cells, and there are some specific type of drugs. Narrow spectrum drugs attack only a few different pathogens, and most of the time this is actually the preferred way to treat a pathogen, because this will lead to less drug resistance that we are going to talk about. On the other hand, if the exact cause for the disease is not known, then sometimes these broad spectrum drugs are used, which attack many different kinds of bacteria, but this is not the preferred kind of treatment in the first place. We always start with a narrow spectrum and then go into the broad spectrum if the pathogen is not identified or difficult to identify in clinical conditions. Then 2 words, the cidal agents, we talked about that, kills the target pathogen. So for example, a bactericidal will kill the bacteria, and the static agents reversibly inhibits the growth of the microbe. What we mean by reversibly inhibiting the growth of microbe is a bacteria, say for example, if it is a bacteriostatic agent, as long as that particular drug is present in the system, the bacteria will not grow. Once you eliminate the drug, the bacteria can grow reversibly. But sometimes there are some antibiotics which are also static agents. They are used to inhibit the bacterial growth, and your own immune system remove all the remaining bacteria. So even if an antibiotic is not cidal, still being static actually do work or get the remission from the disease. So next we are going to talk about that how we measure the effectiveness of antimicrobial drugs, particularly say when we're developing a drug. And the effect of an agent may vary with the concentration, microbe, host in both. So the effectiveness is expressed in 2 ways. One is what we call as a minimal inhibitory concentration, or MIC. So MIC or MIC. So MIC is the lowest concentration of the drug that prevents the growth of the pathogen. There is also another word, which is minimal lethal concentration, or MLC. This is the lowest concentration of the drug that kills the pathogen. So there is a little bit, one is a cidal effect and one is a static effect, prevents the growth. So these are the two differences in the effectiveness. And there are ways that we can measure this. So let's take a look at that. So in the next section, what we are going to do, that we are going to understand this MIC or the MLC, and how do we do this by different tests. And these different tests, the first one we're going to talk about is the dilution of susceptibility test for MIC and MLC. Then there is another test called a disc diffusion test done on an agar plate. This is also known as a Carbibauer method. And then also to determine the minimal inhibitory concentration, this is known as an E-test. This is a commercially available test, and we're going to talk about this as well. So the first thing that what we do in the dilution test, this involves inoculating the media containing different concentration of the drug. So both broth and agar with the lowest concentration showing growth is the MIC. So basically what you do, that you take different test tubes. Each of these test tubes contain a known microbe of a specific concentration. And then you add to that a specific concentration of drug from low to high concentration. And then you check back that which concentration of the drug could inhibit the growth of the microbe. This can be done in a broth as well as an agar plate where you look at the colonies. So you need to identify the agar plate where there is no colonies and identify that specific concentration of the drug, and that will be the minimum inhibitory concentration. If broth is used, tubes showing no growth can be subcultured into a drug-free medium, and broth from which microbe cannot be recovered is the minimal lethal concentration. So there is a subtle difference. So what we did, that for the minimal inhibitory concentration, we are just looking at the tubes or the plates where we have added the antibiotic, and we are looking at the one with where there is no growth of the microbe, because we inoculated with a microbe each one of them, and we are only going to see that where there is no growth. Now for the other purpose, what we do, so from all these dilutions, we do take a little amount of the inoculum, we add that to a fresh media, and which is drug free of course, and we look for microbial growth. If the microbes can't be recovered, that is the minimal lethal concentration in the particular tube from which the bacteria was taken. This is how you can identify the MIC or the MLC from the dilution susceptibility test. The next one is a disc diffusion test, where there are paper discs impregnated with specific drugs at specific concentrations are placed on the agar plates inoculated with a test microbe. And the drug diffuses into the disc, into the agar, establishing a concentration gradient. And we can observe some clear zone or no growth around the disc, and we can identify that which is a more effective antibiotic. So let's take a look at that, how we exactly do this. So this is a Carbibauer method. This is a standard method for the disc diffusion test. So what we do here is the sensitivity or the resistance is determined using the tables relating to the zone of diameter of the microbial resistance. So let's first take a look at how we actually do this. So first we take a Petri plate and we put a microbe of our interest, which could be any one kind. Say for gram positive, if you are taking Staphylococcus aureus, it is Staphylococcus aureus. If you are using Pseudomonas aeruginosa, it is Pseudomonas aeruginosa. And you make a lawn on this plate first. So basically how you make a lawn, you can just put a little bit of bacteria, and with a spreader, you spread the bacteria all around. And this is basically going to grow over the period of time into a lawn if there was no antibiotics. Now, this is an applicator where you have 5 of this, 1, 2, 3, 4, 5 of these discs, which are loaded with different antibiotic, different concentrations into that. And you put this one on top of the plate and press that plunger, and these discs will be put onto the agar plate. Now you allow them to grow for 48 hours or 24 hours. And then what is going to happen, what you are seeing in this particular plate, that around this one, there is no zone of inhibition. This is the background bacteria that is making a lawn in the background. Whereas you can see that around this, there is a small zone of inhibition. Then the next larger zone is this one, then the next one, and this does have the largest zone of inhibition. Now you measure the diameter of each of those zones and you tabulate those. So what you do is that right here, what we are looking at is the relationship in between the minimal inhibitory concentration, MIC, of a hypothetical drug and with the size of the zone around the disc which microbial growth is inhibited is plotted to predict the drug effectiveness. As the sensitivity of the microorganism of the drug increases, the MIC value actually decreases and the zones grow larger. So suppose the drug varies from, say, for example, from 7 to 28 milligram. So in between this, say 7 to 28 milligram per ml in the body during the treatment. So this figure shows that any pathogen with a zone of inhibition less than 12 millimeter, right here, less than 12 millimeter will have an MIC value greater than 28 milligram and will be resistant to drug treatment. A pathogen with a zone of diameter right here, if you look at greater than, say, for example, 17 millimeter, will have an MIC less than 7 microgram per ml and will be sensitive to the drug. So the zone diameter in between 12 and 17 indicates the intermediate sensitivity and usually signify resistance, whereas anything which is above this level, which is above the inhibition zone of 17 millimeter, this is basically the sensitive concentration of the particular antibiotic. So this is how the Carbivore method, using the diameter of the zone of inhibition, is plotted. We can just measure the effectiveness of this drug. Now, this do have some limitations, because think about that some of the drugs may diffuse through this agar plate at different rates. So you really cannot concentrate, or you really cannot identify or compare many different drugs in these same ways. On the other hand, there are some other ways that we can determine this, or the minimal inhibitory concentration, which is known as an E-test. This is actually convenient for use with anaerobic pathogen. And this is similar to the disc diffusion method, but uses a strip rather than a disc. So this E-strip contains a gradient of an antibiotic. And essentially what you are seeing, that this is the highest concentration, this is the lowest concentration. Here the E-strip is in reverse. So here you have the highest concentration, whereas you have a decreasing concentration towards this. And you are seeing that there is an elliptical zone being formed right here. What you mean by this elliptical zone? That means, so this is basically the background, and this is the colored reaction, that if there is no growth, this colored reaction actually happens. So basically this is a hemolytic bacteria. You do not have to go into the detail. So the original color of the plate was uniformly red due to this blood agar plate. And when there is no growth of any bacteria, the color of the blood remained right here. So there is no growth. This is like the zone of inhibition, similar, but just elliptical. Whereas in the background, what you are seeing, that there is growth of the bacteria and clearing of these zones, or hemolysis happened. So what you are seeing, that as you are going from this top to bottom, the zone of inhibition is coming down to a level that up to this concentration, there is no growth. And then beyond that, there is growth of the bacteria. So this is the minimal inhibitory concentration. And here you can see the duplicate. This is the minimal inhibitory concentration. So commercially, you can identify using an E-test an efficacy of an antibiotic. So these are some of the methods that how the efficacy of an antibiotic is tested. So now what we are going to talk about, mainly first talk about the antibacterial drugs, and compare the antibacterial drugs, correlate their lack of microbial growth with selective toxicity, relate the side effects, and explain relative effectiveness. So the first thing that what we are going to talk about, that what are the different types of antimicrobial drug, and what are their main mode of action. So again, remember that the selective toxicity is very important, and how we achieve this selective toxicity for the antibacterial drug. So for the antibacterial drug, the first group is the inhibitor of cell wall synthesis. So in the human being or the host, there is no cell wall. So all inhibitors of cell wall synthesis are very, very selectively toxic. The next one is the protein synthesis inhibitor. Remember that the bacterial ribosomes are very different from the eukaryotic ribosomes. So the eukaryotic ribosomes are made up of 40 and 60S subunit, which makes an 80S ribosome, whereas the ribosomes of bacteria are made up of 30S and 50S subunit, which makes a 70S ribosome. So if we can specifically target some of these drugs, particular bacterial ribosomes, they are selectively toxic. But on the other hand, it is not so much selectively toxic compared to the inhibitor of the cell wall synthesis, because the mitochondrial ribosomes are very similar to the bacterial ribosomes. And these drugs may have some side effect because they can bind to the mitochondrial ribosomes. Then there are some other antibacterial drugs which are metabolic antagonists. They disrupt some of the bacterial metabolism and also selectively toxic, but usually most of the time resistant quickly grow. Also, there are some specific drugs which inhibit the nucleic acid synthesis. Now, the nucleic acid synthesis by RNA-DNA polymerases are pretty similar in between the eukaryotic and the bacterial system as well. So selective toxicity is relatively difficult to achieve. So we do not target, most of the time, we do not target DNA or RNA polymerases, although there are some exceptions, but we try to target some of the drugs to certain other kind of enzymes which are related to the nucleic acid synthesis, and they significantly differ from the counter enzymes which are found in the eukaryotic system. So that's how we can achieve the selective toxicity. We're going to talk about that. So first, take a look at the inhibitors of cell wall synthesis. And the first group of drugs is penicillins. So these, most of them are known as aminopenicillinic acid derivative and differ in the side chain attached to the group. So the most crucial feature for all of these molecules is the beta-lactam ring. This is the most important one. All of them do have a square ring, we are going to see in a second, that contain a beta-lactam ring. This is essential for the bioactivity, but unfortunately, there are many penicillin-resistant organisms that produces a beta-lactamase, which is also known as penicillinase, which hydrolyzes the bonds in that ring. As soon as that beta-lactam ring is lost, the antibiotic loses its effectiveness. So here, what we are looking at, first take a look at different kinds of antibiotics and their mode of action. So it blocks the enzyme that catalyzes the transpeptidation or formation of cross-links in between the peptidoglycan. If you remember that when we have this pentapeptide, you have the N-acetylglucosamine and N-acetylmuramic acid. From this N-acetylmuramic acid and N-acetylglucosamine, which is known as the NAM and the NAG, you have these pentapeptides coming out. And the terminal two amino acids in these pentapeptides are D-alanine and D-alanine attached to each other. Now, these two terminal D-alanines will cause a transpeptidation to the chain to the next and form a cross-linkage in between, whereas one D-alanine will go out. But this cross-linking or the transpeptidation cannot happen. So penicillin inhibits this transpeptidation reaction. So what happens? The cell wall structure falls apart. It cannot form the cell wall, and that will lead to the lysis of the cell because the cell wall is not properly synthesized. So most of the time, these antibiotics are effective when the bacteria is synthesizing new peptidoglycan. That means they are growing in the log phase, and at that time, they are the one which are most effective at that point of time. So now let's take a look at these different types of penicillin. So the first one is the penicillin G, and this is a high activity against most of the gram-positive bacteria, but low against gram-negative. So they are very specific. But there is a problem with this penicillin G, that it is destroyed by the acid, and also by the penicillinase, which many of the particular bacteria produce to destroy these antibiotics. On the other hand, then there is another one, which is called the penicillin 5. And this penicillin 5 is the same spectrum, but they are more acid resistant compared to the penicillin G, and you can orally give this to the people. Then you have another same group of penicillin-based antibiotic, which is known as the nafcillin. This is active against the staphylococci that produce particularly the beta-lactamase enzyme. So this is to some extent resistant to the beta-lactamases or penicillinases, which penicillin 5 was susceptible to. Then we are looking at the next one, ampicillin, which is active against gram-positive. Now it is an extended spectrum. It is also effective against gram-negative bacteria too, and this one is acid stable. Then we have another of the same, which is the carbanicillin. This is effective against the gram-negative bacteria such as Pseudomonas and the Proteus. This is also acid stable, but the problem is that it is not well absorbed by the small intestine. So that's one of the problems. Then you have the next one is the piperacillin. This is an extended spectrum which is active against the gram-negative bacteria along with the gram-positive, including the E. coli, Pseudomonas, Enterobacter, Proteus, and Klebsiella. So you can easily appreciate that how the modification of just the side chains cause this. So if you look at the structure, you can easily appreciate that all of these basic structure of the penicillin do contain this beta-lactam rings. So the penicillinase or beta-lactamase, which give the penicillin resistance, they break this bond right here and open this ring. If this beta-lactam ring is destroyed, then the antibiotic is no good. It loses its effectivity. So what you are seeing that this is the side chain, and all these different ones, different types of side chains, they change the particular properties of these different antibiotics. So you can easily appreciate right here is the larger, bulkier chain, and it is relatively difficult for particularly beta-lactamase enzymes to destroy these because they sit tightly into a particular area of the penicillin binding proteins. Let's take a look at how these work in a little bit more detail. So the penicillin binds to periplasmic protein, and these are known as the penicillin binding proteins. And these happen in naturally occurring penicillin, and penicillin 5 and the G are narrow spectrum. We talked about that. Whereas the others that we just talked, those are the semi-synthetic penicillin, and they do have a broader spectrum than the natural occurring one. So semi-synthetic penicillin means that the basic structure is derived from the original penicillin, but the side chains are added by the chemical reactions in the laboratory. And the bulkier side chains make it more difficult for the beta-lactamase enzyme to degrade that we just talked about in the previous slide. But the problem is that resistance to penicillin includes semi-synthetic analogs continues to be a problem. On the other hand, these aminopenicillins have a broader coverage that includes many gram-negative bacteria that we have just seen right here. But there are some other ways that we can use. There are some specific drugs which is known as a clavulanic acid. The clavulanic acid can inhibit the beta-lactamase. Sometimes if we do think that there is a chance of some resistance going to happen, if there are some penicillin-resistant bacteria, we can add the clavulanic acid along with the penicillin or amoxicillin or ampicillin and administer it to the patient. A very common thing that you probably heard about that all the time is known as augmentin. This augmentin is a common drug that is given to many of the ear infections in oral suspensions, particularly the children. And this augmentin is essentially a combination of this amoxicillin along with this clavulanic acid, which is basically an inhibitor of this beta-lactamases. Once the clavulanic acid inactivate the beta-lactamase, the penicillin-based drugs can work its proper way. So here is a beta-lactam antibiotic block this transpeptidation, and we just talked about that right here is a NAG, N-acetylglucosamine, and this is your N-acetylmuramic acid, and that's the pentapeptide. And you have two terminal D-alanines. And here you are seeing that this is the transpeptidase subunit, which is also known as a penicillin binding protein. And this transpeptidation reaction, what will do that it will make a bond in between these two peptides, and this is basically the process. In this process, one of the terminal D-alanine moves out. So here it remains as 4 peptides. Here it is 5 peptides. So the third amino acid is attached to the fourth amino acid of the NAM. This is how the cross bridge forms, which is known as a transpeptidation reaction. Now, what happens is that you can easily see that penicillin specifically bind to particular this area, which can be penicillin, cephalosporin, monobactam, carbapenem. These are all different types of beta-lactam, and all of these antibiotics, we just talked about the penicillin, but we are going to talk about the cephalosporin, monobactam, and carbapenem as well. All of them binds to the site of this penicillin binding protein or the transpeptidase and blocks its active site. So once the active site is blocked, this transpeptidation cannot happen, and layers of this peptidoglycan cannot be attached to each other, which causes the cell wall to fail, basically, and the cell wall will not be properly put together, and the cell will be lysed because it doesn't have the cell wall outside its plasma membrane, and it can easily lyse in the aqueous environment. So the next one, what we are looking at is the cephalosporin. These are structurally and functionally very similar. These are broad spectrum antibiotic and can be used by most patients that are allergic to penicillin. There are 4 spectrum of these activity, and we call this as a first, second, third, and fourth generation. Cephalosporin is in the market for a while, but there are 2 others that we talked about in the previous slide, which is the monobactam and the carbapenem. These 2 drugs were developed in the 1980s, but unfortunately, there are resistance for each of these drugs. The next drug, which was also considered as a magic bullet, was vancomycin. And this vancomycin is produced by the Streptomyces orientalis. And you can easily see that this is a kind of a cup-shaped molecule. And this is a glycopeptide antibiotic. And this also inhibits cell wall synthesis. But the function is a little bit different compared to the penicillin binding proteins like your penicillins, cephalosporins, carbapenems, or the monobactams. So what happens that this vancomycin doesn't bind to the penicillin binding proteins. Instead, they bind to those pentapeptides at the 2 terminal alanines, D-ala, they bind to that region. As vancomycin blocks those 2 terminal alanines, the transpeptidation is basically blocked. And this is also very important for the treatment of the antibiotic, which are resistant to staphylococcal and enterococcal infections. But the problem is that the resistant staphylococcal enterococcal infections is from the penicillin. So once penicillin doesn't work, you actually work on these vancomycin. So previously, this was thought about that this is a drug of the last resort, but unfortunately, there are resistance. We have vancomycin-resistant Staphylococcus aureus, which are known as a VRSA, vancomycin-resistant enterococci, which is known as a VRE, and these are some of the great concern for these 2. So the basic difference, again, just to remember that in between the penicillin group and the vancomycin is that penicillin inhibit the enzyme, which is the transpeptidase, or also known as a penicillin binding protein, which catalyzes the transpeptidation reaction. Penicillin sits in the active site of that, mimicking the structure of the 2 terminal D-alanines. On the other hand, the vancomycin do not inhibit that enzyme. Instead, or they do not sit into the active site of the enzyme. Instead, they bind to the 2 terminal D-alanines on the pentapeptide, which is attached to the N-acetylmuramic acid, and doesn't allow the enzyme to access the substrate. And that's how they can stop this reaction. So the next group of antibiotics are the protein synthesis inhibitor. And many antibiotics bind specifically to the bacterial ribosomes and that target different steps of protein synthesis, which could be amino acid tRNA binding, peptide bond formation, mRNA reading, translocation. These are the different steps to which these antibiotics are targeted. So the first one is the aminoglycoside. So this aminoglycoside is a large group, all with a cyclohexane ring. So right here, you can see a subtle cyclohexane ring right here, cyclohexane ring, which is added with these amino sugars. So you can see that you have an amino sugars right here, amino sugar, you have an amino sugars. So they bind to the 30S ribosomal subunit and interfere with the protein synthesis. And the way they do it, that these cause the misreading of the messenger RNA by the ribosome. And there's a large group of antibiotics in this class. You definitely heard about a good number of them. Say, for example, you have streptomycin, but along with that, you have kanamycin, neomycin, tobramycin. These are all derived from different species of the Streptomyces bacteria. On the other hand, there's one which is known as a gentamicin. Gentamicin is not from the genus Streptomyces, but it is derived from a different bacteria, which is known as the Micromonospora purpurea. The streptomycin, on the other hand, was classically used as a TB drug, but it is a little bit toxic and may have side effects, because we already talked about that there are ways that these drugs can bind to mitochondrial ribosomes, which are similar to the bacterial ribosomes, and cause problems. So do have some toxic side effects. But again, resistance also increases, and the streptomycin is not used anymore for the treatment of TBs. All tetracyclines have a 4-ring structure to which a variety of side chains can be attached, and they can be named accordingly. Say, for example, a chlorotetracycline with a chloride right here. The commercial name for the chlorotetracycline is oreomycin, which consists of a tetracycline with a chlorine atom. On the other hand, the doxycycline, which contains a hydroxyl right here. These are all broad spectrum, but they are bacteriostatic. They target the 30S subunit of the ribosome by inhibiting the protein synthesis as well. The next group of antibiotics are known as the macrolides. Macrolides do contain this 12 to 22 carbon lactone ring linked to 2 or more sugars. And a very good example for this is an erythromycin, which is a broad spectrum, usually bacteriostatic. And this binds to the 50S ribosomal subunit instead of 30S. The last 2 examples that we saw is a 30S binding and misreading of the mRNA, but instead the macrolides bind to the 50S ribosomal subunit and inhibit bacterial protein elongation. And the usage of these started. Some of the patients who are allergic to the penicillin are prescribed macrolides. So these are great against gram-positive bacteria, mycoplasma, some gram-negative bacteria as well. So these are treated for some of the very difficult disease like whooping cough, diphtheria, diarrhea by Campylobacter jejuni, then pneumonia from Legionella. We talked about this Legionella in a different context, which is a cold-loving bacteria, can come from the air conditioning duct, which is called the Legioniosis type of pneumonia, also the mycoplasma. Another drug which is related to the erythromycin is known as an azithromycin. And azithromycin is actually used much more compared to the erythromycin. Azithromycin is also a drug which can cure the chlamydial infection as well. There is also another group which are known as a ketolides. The ketolides are the semi-synthetic macrolides, which actually do have a better acid stability and also oral bioavailability. Tethromycin is one of these ketolides which are used to treat community-acquired pneumonia. Then what we are looking at, a specific group which is known as lincosamines. These are also produced by the Streptomyces. These are broad-spectrum antibiotic against anaerobic microbes, but less effective against aerobes. And this is used sparingly because they can support indirectly the growth of the Clostridium difficile, which can cause a superinfection, which can result in other disease states. So they can kill many of the normal microbial flora in our intestine, and where the Clostridium difficile can take over and cause a superinfection. The clindamycin is an example for this, and this is used to treat dental infection as well as some staphylococcal and streptococcal infections. So the next one is the chloramphenicol, which is isolated from Streptomyces venezuelae, but it is now chemically synthesized. It binds to the 50S ribosomal subunit and inhibit the bacterial protein synthesis. But it is toxic to some extent with numerous side effects, and so it is only used in life-threatening situation. But there are some, like it is used in the eye drops but they are not used systematically because the eye drops is still considered as topical and do not have a systemic side effect or toxic side effect. But this drug is hardly used as a systemic effect, which is orally or intravenously. The next group of antibacterial is known as these oxazolidinones, which is a synthetic drug with heterocyclic 5-membered ring structure. These are mainly bacteriostatic, and they inhibit the protein synthesis by binding to the 23S ribosomal RNA within the 50S ribosomal subunit before the protein synthesis has begun, and preventing the assembly of the 70S initiation complex made by the 50 and the 30S. So this is the drug which is called linezolid. And the linezolid is one of the rare antibiotics that was discovered in the 21st century. And this is active against the methicillin-resistant Staphylococcus aureus, which methicillin is a penicillin group of drugs. Then they are good against the vancomycin-resistant Enterococcus, penicillin-resistant Streptococcus pneumoniae, and many of the gram-positive anaerobes. But the linezolid is not prescribed at home. Most of the time, linezolid is used in the hospital setting because this is one of the drugs that the doctors want not to be out in the outside so that there is some resistance. These are only used in very serious conditions in the hospital settings. The next group of antibacterial drugs, which are known as the metabolic antagonist. These drugs act as antimetabolites. They antagonize or block the functioning of metabolic pathway by competitively inhibiting the use of metabolites by some of the key enzymes. These are usually structural analogs, and these molecules are structurally similar to compete with the naturally occurring metabolic intermediate to block the normal cellular metabolism. Unfortunately, drug resistance also occurs with these drugs as well. So here we are looking at the structurally related compound, which is known as sulfanilamide. Sulfanilamide, which is also known as the sulfa drug, is very structurally similar to para-aminobenzoic acid. So it is an analog for the para-aminobenzoic acid. Now this PABA or para-aminobenzoic acid is used for the synthesis for the folic acid, and which is made by many pathogens. And the folic acid is a vitamin. We have to get it, the human beings need to get it from the environment or from the outside source. But for the bacteria, they produce this folic acid, and this is extremely important to make the nucleotides finally. So once the drug inhibits the enzyme by competitive inhibition, the bacteria fails to produce this folic acid, and then the nucleic acid synthesis doesn't happen, and that's the mode of action. So what you are here seeing that this is the chemical structure of a dry hydrofolic acid, or known as DFA. And this is a natural substrate for an enzyme which is known as the DHFR, or dihydrofolate reductase. And this enzyme is in the folic acid pathway. Trimethoprim is a synthetic antibiotic that also interferes with the folic acid production. So what you are seeing right here is that this is the structure of the trimethoprim, and it is in red here. So what this structure shows that this is the chemical structure of the trimethoprim, and it is sitting at the active site of the enzyme, which is the dihydrofolate reductase. So this binds to the active site of the dihydrofolate reductase and stops the activity of the enzyme, blocks the folic acid synthesis pathway, just very similar to the sulfa drugs. The next one is a nucleic acid synthesis inhibition. The most commonly used antibacterial drugs that inhibit nucleic acid synthesis function by inhibiting DNA polymerases, which are fluoroquinolines and topoisomerases. And also there are some which is known as a rifamycins. But these drugs, again, as I said before, are not as selectively toxic as other antibiotics because bacteria and eukaryotics do not differ greatly in the way they synthesize these nucleic acids. So the first one, what we are looking at is a fluoroquinoline. So fluoroquinoline is a synthetic drug which do contain these 4 quinoline rings, and they act by inhibiting the bacterial DNA gyrase, which is responsible to give a negative twist to the DNA and separating them during the replication. And also they inhibit the bacterial topoisomerase 2, which is responsible for untangling the bacterial DNA during the replication from the supercoiling. So both of these directly act on the DNA replication. These are broad-spectrum bactericidal, treat a wide variety of infections, and one of the common name for this is a ciprofloxacin, is one of the fluoroquinolines. The next part that we are going to talk about is the antiviral drugs. They are much, much less compared to the number of antibacterial drugs. So let's take a look at these antiviral drugs. It is slow because it is difficult to specifically target the viral replication. And antiviral drugs had mixed success, and the vast majority of viral infections cannot be cured, and it has to rely on your own immune system for the cure. And some antiviral drugs simply limit the duration of the illness. It doesn't cure it. Say, for example, for flu, or the severity, for example, herpes, HIV, chickenpox, or shingles. The antiviral drugs, which are mostly currently in use, inhibit the virus-specific enzyme and inhibit with the life cycle process. So before we go into the detailed specific of that there are some specific drugs that were developed. You will see that it is discussed in the last part of your antiviral drugs in your textbook, that interferon alpha and ribavirin, these were some of the classically used antiviral which were used for a long time. But the problem is that what happens, it makes people extremely sick by the interferon alpha treatment, and ribavirin treatment do have a huge amount of toxicity. Although interferon alpha is used in some of the viral infections when there is a need, ribavirin usage has gone down specifically. There are some new development that is very exciting, and this happened in a couple of years back, 2013, 14, 15, that the drug for treating the hepatitis C virus has been discovered, and sofosbuvir is a nucleotide analog which is designed to treat hepatitis C virus. Along with that, there are two other drugs which is known as the ledipasvir, and another is known as velpatasvir. So usually a combination of these two drugs out of the three are used in different type of HCV infection. Now, the hepatitis C virus, you will see that this is all in your textbook. There are mainly in the United States, the HCV type 1 or the genotype 1 is the most common one. And for that, we usually use a combination of sofosbuvir and ledipasvir. But on the other hand, mainly for the type 2 or 3, which is common in other parts of the world, the prescription is usually the sofosbuvir and velpatasvir. So these combinations actually do have some effect in different genotypes of this hepatitis C virus. So let's first take a look at some of these antiviral drugs which treat influenza, a Tamiflu, estelmavir, and this is a neuraminidase inhibitor. And this is an anti-influenza drug. And this is not a cure of influenza, and this has been shown to shorten the course of illness, but you have to administer this drug within about 48 hours after you get the flu infection. The way how it works, so when the influenza virus actually buds out of the cells, at that time, it is attached onto the plasma membrane by some specific glycoprotein. And what is needed, that you need these neuraminidase enzymes where this a bond in that those glycoproteins are severed so that the virus is released from the plasma membrane surface. If this neuraminidase is inhibited, the virus is failed to come out or separate from the cell that it's coming from. So all these viruses are stuck on the plasma membrane, and eventually they will be killed by your immune system. So that's how the antiviral drug Tamiflu or estelmavir works. There are some other drugs in the market as well, which is known as the Peramivy, which is an intravenous injection. Also another drug which is known as zanamivir, which is also used with an inhaler as well. Those are your anti-influenza drug. So the next what we are looking at, these are the drugs that we are going to talk about, is the antiviral drug with viruses with DNA genomes. And these are also known as the nucleosides analogs, and they basically cause chain termination. When they are incorporated into the DNA chain, the chain no longer can extend, and that's how it fails the viral DNA replication. Good example for these nucleoside analogs are the acyclovir, and this vidarabine is an ointment that is also used. These are used to treat for herpes virus infection, as well as chickenpox, which is a varicella zoster virus, which is also a herpes virus, shingles, the same virus, and this is an ointment that is also used. On the other hand, what you can see, this ganciclovir is used to treat the systemic cytomegalovirus illness, which is also DNA virus. On the other hand, you can see the foscarnet is like a pyrophosphate analog, and it treats the illness caused by both herpes simplex virus and cytomegalovirus, which are all DNA viruses. Then you have a broad spectrum antiviral drug, which is known as cidofovir. This inhibits viral DNA polymerases. This is a mononucleotide analog used particularly in viral eye infection or retinitis. Cidofovir is one of the example of viral DNA polymerases inhibitor. So here we are looking at the life cycle of the HIV, and we are not going to go into the virology of the HIV, but we are just trying to identify the different areas where these drugs do work. So here what you are looking at, that this is the particular HIV, and on the cell surface you have these particular molecules, which are the CD4s. And the viral, there are some specific viral glycoprotein GP120, GP41, which do lodge with the CD4, and then it fuses with the plasma membrane. So one group of drug inhibits this fusion, and these are known as fusion inhibitors. So this will not allow to fuse the virus with the plasma membrane. Then what happens? The next part right here, the plus-stranded genome, positive sense RNA genome, comes out into the cell, and the virus do contain the reverse transcriptase, and this reverse transcriptase will make it into the DNA. So the reverse transcriptase inhibitors block this step of converting the RNA into the DNA. So that is your reverse transcriptase inhibitor. The next part, what happens a little bit later, let me come back to this later. But what happens after the process of making the DNA from the RNA by the reverse transcriptase, although this is blocked by the RTs, this made up DNA goes into the nucleus and it will bind or recombine with the host chromosome. And to recombine with the host chromosome, there is an enzyme that is responsible, which is also a viral enzyme, which is integrase. So there is some integrase inhibitor, which blocks the integration of the viral DNA, which is converted from the RNA to the viral DNA, and integration into the host chromosome. Suppose a person is already HIV positive. So for that person, the portion of the HIV DNA is already in the genome, and it makes the copies of the mRNAs. The mRNAs synthesize all the proteins, and all these proteins are required for the virus to assemble. Now, these proteins, or the HIV proteins, are produced in a form which we call as an HIV polyprotein. So these proteins are attached from one after another, N-terminal to C-terminal. The C-terminal of the first protein is attached to the N-terminal of the second one and the third one. So these are known as the polyproteins, which are chopped into individual protein by an HIV protease. So this protease inhibitor blocks this step. So these viral polyprotein cannot be chopped into each individual HIV protein. So they cannot assemble into the virus and bud out of the cell. So these are some of the targets of the anti-HIV drugs. So let's take a look at that. So first thing what we are looking at is the nucleoside reverse transcriptase inhibitors, or in short, it is known as the NRTIs. This target interferes with the critical step of the viral replicative process or inhibits the reverse transcription. Azithromycin is an example of this. The commercial name is also, there is another similar drug which is known as the zidovudine. but these are not used that much due to high amount of resistance. Then you have the protease inhibitor. We just talked about that. This blocks the activity of the HIV protease needed for the production for all viral proteins. Then this is the structure of this ritonavir, which is the protease inhibitor. This blocks the activity of the HIV protease, which is needed for basically production of all viral proteins from the polyprotein. It cannot chop up the polyprotein to make the individual protein. So that's your viral protease inhibitor. The next we are looking at is a non-nucleoside reverse transcriptase inhibitor, or NNRTIs. These prevent the HIV DNA synthesis by selectively binding and inhibiting the viral reverse transcriptase enzyme. So it binds to the specific active site of the enzyme by blocking the enzyme. Then we talked about the integrase inhibitor, prevent the incorporation of HIV genome into the host chromosome. So the fusion inhibitors prevent the HIV entry into the specific host cells, so the receptors cannot properly bind to the viral spike proteins and induce the membrane fusion. And most of these HIV drugs are not just one drug, but usually it's a combination of different drugs, 2 or 3 different drugs. The reason for that reduces the chances of getting resistance. Then we are going to talk about some of the antifungal drugs, and those are much fewer effective agents because of the similarity of the eukaryotic fungal cells and the human cells. And many have low therapeutic index and are toxic, and these are easier to treat superficial mycosis on systemic infections, because the problem is that you can use some of the relatively toxic products on your skin surface, but systematically administering them is difficult due to the toxic effect. And sometimes some combinations are used. So the mycosis is basically the disease which is caused by the fungus. And there is superficial mycosis, and the superficial mycosis usually happens on the skin. For example, Candida albicans is one of the fungus which can cause this superficial mycosis. It can also be cutaneous, like on the skin, but it can also be subcutaneous as well, or under the skin. And for these, we use usually these pollines or azole group of drugs that we're going to talk about in a little bit detail, or look at the structure in the next slide. And usually the treatments are the topical, or also in oral medication can be taken as well. So what these drugs do, they disrupt the membrane permeability, and also they inhibit the ergosterol synthesis. So what is ergosterol? If you recap, all animal cells do have cholesterol in their membrane. So instead of cholesterol, there is an interesting sterol in the fungus, which is known as an ergosterol. The synthesis of ergosterol is inhibited, the membrane integrity is completely lost. They also disrupt the mitotic spindle and may inhibit protein and DNA synthesis. On the other hand, the systemic mycosis is much more difficult to control, and it could be fatal as well. And this is much more common in the AIDS patient. And for these, there are 3 common drugs. One is an amphotericin B. This again binds to the sterol and disrupts the membrane integrity. There is flucocytosine. This disrupts the RNA function in the fungus, as well as there is fluconazole with a low side effect and used mainly prophylactically as well. So here we are looking at some of these specific drugs. So these pollines do bind to the sterols, as well as this nystatin also binds to the sterol, and that disrupt the membrane function. On the other hand, what happens, the azoles do inhibit the membrane permeability and include the ketoconazole or miconazole. So the drug, which is known as griseofulvin, this do disrupt the nucleic acid synthesis, protein synthesis, as well as cell division. And then you can see that flucocytosine that disrupts the RNA function. So these are the common antifungal drugs. So the mechanism of drug action for many antiprotozoal drug is not precisely known. Many of this do work by producing some of these reactive oxygen species or reactive nitrogen species that we are going to see. Interestingly, some antibiotics inhibit some bacterial protein synthesis or used against protozoa as well. On the other hand, there are some specific drug which is known as the chloroquine or the mefloquine. These are used for malaria. Another drug which is known as artemisinin, that is also another drug for the malarial parasite, which is Plasmodium. So the exact mechanism of this is not known, but it is thought that the artemisinin actually do produce these reactive oxygen species and disrupt the electron transport chain, and they are responsible for the hemoglobin catabolism disruption, and then finally reactive oxygen species kill the protozoa. Then there is a metronidazole. This produces some nitrogen oxidative damage or reactive nitrogen species that are used for the entamoeba infection. There are some other drugs that is mentioned in your textbook as well, which is known as the nitrazocyanide, which is useful in Giardia infection and Cryptosporidium infection. Both of them can lead to gastrointestinal disorders. But another interesting part that, as I said before, some aminoglycoside antibiotics like clindamycin, petramycin also can be used against some of the protozoa, which are known as the epicomplexan protozoans, which do have an epicoplast, and this epicoplast is a derived organelle from the plastid, and they bind ribosomes, or this clindamycin or petramycin binds to these ribosomes, to these organelles, and inhibit protein synthesis. These are effective against Toxoplasma gondii, against Cryptosporidium. They are good against Plasmodium as well, but it is not one of the main therapy that is used. Chloroquine, mefloquine, or artemisinin are more common drugs for the malaria. For Toxoplasma treatment, there are also some very specific drug other than these clindamycin, which is known as this pyrimethamine, and then you have dapsone and also sulfadiazine. So these drugs are also known as sulfa drugs, which is similar to trimethoprim, which inhibits the folic acid pathway by inhibiting the dihydrofolate reductase or the DHFR enzyme. So the last part we are going to talk about that what is the concern about the antimicrobial drug resistance and what is the public health threat. So one of the type of drug resistance is intrinsic, which means that the mycoplasma are resistant to beta-lactam antibiotics and other cell wall inhibitors simply because these bacteria lack cell wall. On the other hand, their second type is acquired. This occurs when there's a change in the genome of a bacterium that converts it from one that is sensitive to an antibiotic to one that is resistant. On the other hand, the drug-tolerant bacteria, which are called the persisters, lack the mechanism for antibiotic resistance and ignore the presence of antibiotics, usually because they are embedded in biofilms that antibiotics cannot effectively penetrate or are growing too slowly to be inhibited. The importance is that why it is very alarming, because there is a growing drug resistance and these species are appearing everywhere every day. We talked about the methicillin-resistant Staphylococcus aureus, MRSA. Then after that, we thought vancomycin is great, but there is a VRSA, vancomycin-resistant Staphylococcus aureus. There's vancomycin-resistant Enterococci. Then we thought there's a carbapenem or monobactam. Say, for example, carbapenem discovered in the 1980s, which is a late discovery. And we thought that all the different penicillin-based drugs which are susceptible to the beta-lactam antibiotic, carbapenem is the magic bullet for that. But the CRE or the carbapenem resistance enterococci, which is known as a superbug, is a great threat to this antibiotic resistance. So the scientists are constantly trying to find different ways to do this. So before we go into this, let's take a look at how we look into these different ways or how the bacteria develop this antibiotic resistance. So the first way that it does is by preventing access or altering the target of the antibiotic. Say, for an example, what happens that we talked about this transpeptidation reaction as well, and the penicillin binding protein's active site is occupied by the beta-lactamase. Now, if that enzyme changes in a shape and then still does the transpeptidation reaction properly, no longer the particular penicillin-based drugs or the beta-lactam antibiotics cannot bind to the active site because the enzyme modified its active site. So this is a way that preventing access to the target of the antibiotic. The same way we talked about that vancomycin binds to those terminal alanines in the pentapeptides, the D-ala and the D-ala. Vancomycin binds to those areas. So if those are changed to different amino acids, and that is done by the bacteria to, say, for example, D-lactate or D-serine, instead of D-alanine, they incorporated these different molecules. At that time, vancomycin cannot bind to those areas, and the bacteria become vancomycin resistant. The second type that how the resistance can occur is degrading the antibiotic. Say, for example, if the bacteria do have the enzyme beta-lactamase, you can directly degrade the beta-lactam antibiotics, which are all the penicillin-based antibiotic and the cephalosporin-based antibiotic. So instead of doing this, there is a compound which is known as a clavulanic acid. This clavulanic acid is added to your drug, which is a penicillin-based drug, and added. Clavulanic acid inhibits the beta-lactamases, and when the beta-lactamases are inhibited, your penicillin, amoxicillin, or any penicillin-based drugs can do their work. On the other hand, you cannot use the carbapenem, but again, carbapenemases or the resistance against that also have arose. So there are some other ways that the degrading of the antibiotic can happen. Say, for example, aminoglycosides are inactivated by acylation of the amino group, or phosphorylation or adenylation of the hydroxyl group. These are some of the ways that the degradation of the antibiotic takes place. Also, they can alter the antibiotic or membrane structure, like changing the porin structure, which are used by a specific antibiotic to enter into the cell. The specific porin structure is a change, and the particular antibiotic will not be able to enter into the cell. On the other hand, there is also another type of resistance, which is the multi-drug resistant pumps. The multi-drug resistant pumps, also known as the efflux pump, they can rapidly pump out different molecules from the cell. And think about this, that many times these drug pumps are found in the outer membrane of the gram-negative bacteria. So when a drug enters into the periplasmic space by breaching the outer membrane, before that drug can get into the plasma membrane, passing through the plasma membrane into the cell, the drug accumulates into the periplasmic space. And from the periplasmic space, this efflux pump immediately transport that drug back into the environment before the drug can reach inside the cytoplasm of the cell. So these are some of the ways that the drug resistance do appear. So the commercial gene expression systems are designed to identify the production of specific resistant factors such as target modifying enzymes. And there are several tests commercially available which do have this color change induced when the chromophore is acted upon by the beta-lactamase or an antibiotic modifying enzyme. So if a patient is coming to the hospital with a particular infection, they can easily test this very quickly to identify if the patient is having an infection with a resistant bacteria or not. And these color changes can be spectrophotometrically observed. Protein concentration is extrapolated from a standard curve, and these are very standard processes. And also there are some detection systems commercially available, and these can use quick polymerase chain reaction or PCR reaction to identify or detect the drug resistance. So the main problem that we are facing, the proper usage of antibiotics is the most important for the drug resistance. Many of the developing countries, there is not much control of the antibiotics. To minimize the drug resistant increase, the human beings, us, we have to be more responsible. There are many developing countries where the antibiotics are not properly used, and this is sometimes due to the lack of the economics. Many of the patients do not complete the course of the antibiotic. When they feel just okay, they just, due to the financial burden, they just quit taking the antibiotics. We do use a lot of antibiotics in the farm, in the animal feed. Those can lead to resistance as well. To minimize the resistance increase, we need to stick to the narrow spectrum antibiotics, and broad spectrum antibiotics should be sparingly used only when there is not a proper diagnosis. So some of the concerns are: give the drug in appropriate concentration to destroy the susceptible microbes and most spontaneous mutants. So usually if 2 or more drugs given at the same time, we minimize the chance of getting a resistance and use only drugs when necessary. That's very important. And some of the future possible solutions are continuing developing new drugs. Many of the biotechnology pharmaceutical companies are working on structure-based drug design for developing new antibiotics. And also, along with the new antibiotics, many researchers and the scientists are working on quorum sensing molecules where the communication system of the bacteria can be completely disrupted. And also, we talked about that in the previous chapter, that bacteriophage to treat the bacterial diseases also could be on the horizon. So this is the overall idea about the chapter. The drugs that control the microbial growth has played a pivotal role in developing healthy societies across the last century or so. The number of deaths due to the infectious disease from the 1920s and 30s drastically dropped. But we need to understand that how these drugs actually do work, and there is a constant evolution of resistance against them by the microbes, and we have to be very vigilant about that, how we can minimize the development of this drug resistance.